Good morning, all. If we could just ask you to take your seats. We're going to get started in a minute. That would be great. Just as I remember this from 3 years ago, it's a very well-behaved audience that follows all the instructions, so thank you. And we have one or two more people just coming in the back door. And if we can get them seated, that's terrific. I'd like to say good morning to everybody. I am Mark Roithmare. I have the privilege and honor of serving as the CEO of the Alzheimer's Drug Discovery Foundation. It has been, first of all, show of hands, anybody who's been to the fall luncheon in the past for us in Symposium? Just, yeah. So it's been 3 years since we gathered in this room, and maybe you noticed a couple of things. One, the Pierre got a facelift. Doesn't it look fabulous, right? Number two, though, and probably the more important thing, much more important thing, there's been a lot of movement on the science in the last 3 years, and I'm just going to give you a couple of tidbits. One, we now know that the warning signs of Alzheimer's start 20 to 30 years before the symptoms start to show. We also know that now we have, in addition to a PET scan that's done by Lilly that can diagnose Alzheimer's, there's now actually a blood test funded by the ADDF, funded by you all, that can diagnose that you're going to get Alzheimer's. 3 years ago, I would have said this, or Dr. Phillip would have said this, and most of you in the audience who said, yeah, but why would I want to know? Can I do something about it? 3 years later, the answer is yes, you can do something about it. We now know that there's prevention and lifestyle preventions that you can do that, excuse me, literally will push out early symptoms of Alzheimer's. One. Two, in the last 3 years, for the first time, we have new drugs that are getting approved that can take away the plaques and tangles that are the telltale signs of Alzheimer's. And some of them are beginning to show that they improve cognition, but not totally. And that's the third part of this. Probably the biggest movement in the last 3 years, the complementary approaches that tackle the underpinnings of what actually causes those plaques and tangles. Genetics, metabolism, inflammation, vascular. There is a cadre of drugs in the pipeline that are looking to attack that now because, you know, those of you who've been with us in the past, I know you always heard, and Leonard and Ronald Lauder would always say this, hope is on the horizon. I will tell you that hope is tangible. We now know there's prevention. We know it will be combined therapies. We know it will be precision medicine. It's getting to the point we can literally almost touch it. With our wonderful panel, Dr. Phillip and the entire panel, you're going to hear about all of this today. Before we start, I have a couple of acknowledgments. The first, to the sponsor of this symposium and luncheon. They've been doing it for several years. They also sponsor us in Washington, D.C., the Eli Lilly Corporation, and Mary Beth is here from Eli Lilly, such a good friend. I'll say this about Eli Lilly. They're not just sponsors. They've been partners for a long time with us, strategic partners, and we look at a hopeful future. They are also a company that we've gotten to know that they are both innovative and passionate in the pursuit of helping people lead healthy lives. So, yay, Lilly, Mary Beth. We'll tell you that after the symposium, there is a luncheon. We have two marvelous honorees this year. One is a longtime friend, Carol Bollinger. Those of us who know her like to say, if you know Carol, you're in awe of Carol. And those of you who don't know her, I promise you by the time you leave today, you will be in awe of her. And then the other is a brand new friend we have, the NFL, National Football League Players Association. I will tell you, we've gotten to know them, Howard and I, in the last year, and I will tell you that their concern for cognition, their concern for brain health is second to none of any organization in the United States or worldwide. You'll hear about that today. They are part of what we call our diagnostic accelerator. This is something 3 years ago when we were together, we talked about the $50 million diagnostic accelerator. 3 years later, it's now a $100 million diagnostic accelerator, thanks to Leonard Lauder, Bill Gates, Jeff Bezos, Mackenzie Scott, Eli Lilly, our good friends from AFTD who are here with us today that have been in on it from the beginning. And this is a program that we're funding, the $100 million, to find blood tests, eye scans, artificial intelligence that can help diagnose Alzheimer's before the symptoms even start. And it's also in pursuit of new and better biomarkers to pinpoint the clinical trials for that precision medicine that I'm telling you is just about within our grasp. Also, just an acknowledgement to the Lauder family. We'll have 7 or 8 members of the Lauder family with us at lunch today, spanning 3 generations. I bring that up because all of you who have given so generously to us today know that because of the Lauder family, everything here today, the food, the service, is all covered by the Lauder family. So we can say to each and every single person here, all your money goes to the scientists that you're going to meet today. So pretty cool, right? Just a little bit of housekeeping before we get into this. One of our panel members had a family emergency, couldn't make it today, so Howard will actually be filling in for Jose Luxinger today. So Howard's going to play multiple roles for us today. We'll also tell you that as I turn this over to Dr. Phillott, it'll be about 45 minutes of presentations that he'll walk you through. Then we're going to break and do 30 minutes worth of questions from the audience. And then at the end of that, so you all have little pads and pens and paper in front of you. Please write your questions down as we go. You'll get a chance to ask them. If for any reason we run out of time and you can't get your question answered, all you got to do is put your name on the bottom of it and hand it to one of our staff. Staff, can we just all raise our hands here? Fabulous staff, by the way. Fabulous staff. Just hand it to one of the staff members, and we will follow up with you post to make sure your question gets answered. Now comes the best part of the entire job that I do here. I get to introduce Dr. Howard Phillott. For over 40 years, and Howard, we've been saying over 40 years about you for probably close to 10 years. I don't know, where, might be for 15 years, but we're just going to say, despite his youthful looks. Over 40 plus years, geriatrician, neuroscientist, he has dedicated his life and soul to the pursuit of preventions and therapeutics and cures for Alzheimer's. To give you a sense of personally, Howard, we've worked together the last 6 years. During these past 3 years, during the pandemic, every single day at 8:45 in the morning, every single day, we FaceTime each other. And we talk about the science, the funding, the biomarkers, the prevention, the therapeutics. And sometimes we agree, sometimes we go back and forth a little bit. But always, 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 Howard has one true north. How is all of this that we're talking about, that we're funding, that we're thinking about, that we're fighting for, how does it help the patient? Because we know Dr. Phillott is both a neurologist and a geriatrician. He keeps the patients as his true north. He keeps the patients as our true north. Inarguably, he's one of the top 5 scientists in the world when it comes to Alzheimer's. Ladies and gentlemen, I give you my good friend, Dr. Howard Phillott.
Thanks so much, Mark. I'll just make a few comments just to sort of frame what we're going to talk about today. We were going to have 3 clinical trial programs presented to you, and I think that's representative of the fact that we can do that today. 10, 15, 20 years ago, we probably would have been talking more about preclinical chemistry, but now we can talk about clinical trials, testing drugs, novel drugs in humans. From the beginning of the founding of the foundation back in 1998, we've had a unique strategy that we've promoted in the field that's finally, I think in the last few years, being adopted by the field. And that's the fact that aging is the leading risk factor for Alzheimer's disease, and Alzheimer's is a disease of old age. And the reason that that's important is because there's been well over 100 years of research on the biology of aging, what causes aging. both in humans and in mice and in all kinds of ways. And the challenge has been, how do we translate that knowledge about the biology of aging into new therapeutics for Alzheimer's disease? And that's what we've been doing for a long time. And I think that also helps us to learn from other diseases, which we'll talk about. You're going to hear about some of that today. And so pulling all that together, it makes this an incredibly exciting time in the field, because we're going beyond looking at just trying to remove the pathologies of Alzheimer's, the plaques and tangles, which were the first avenues for developing drugs. You know, if we could just get rid of the plaques and tangles, everything would be okay. But as Mark mentioned, what we really want to do is understand how and why those plaques and tangles actually happen. And that's where the biology of aging comes in, and that's where new therapeutics come in to reduce inflammation that can cause the plaques and tangles, and so on. And in fact, I think what's really exciting and changed, and I don't know if we can get a little quiet back there. Yeah. Thank you. What's really different is probably 5 years ago, more than half, or maybe even the majority, well over the majority of drugs that were in development were, let's say, anti-amyloid drugs or tau drugs, the tangles. But today, in the last year or so, over 75% of the drugs that are in clinical development are non-amyloid, non-tau drugs. They're novel drugs that address, again, the inflammation, the vascular problems, the epigenetic problems that occur in the disease, and so on. And this is really a revolution. There's about 120 clinical trials going on now in Alzheimer's disease, and I'm proud to say that actually the Alzheimer's Drug Discovery Foundation has contributed to supporting about 20% of the drugs that are currently in clinical trials. And the one amyloid or related amyloid, Lisa will be telling us about in a little while. And so what we envision is kind of like the way cancer has evolved. With aging, many things go wrong in the cell. And if you just try one type of drug, it's like a whack-a-mole. The rest of the things that are going wrong in the cell will come up, and the cancer continues to grow, and we can't treat it. So we need to hit the cell with 3, 4, or 5 different drugs to kill that cancer cell and cure the tumor. And the same thing we believe is going to happen in Alzheimer's. We're going to have to figure out the many ways that neurons get sick and die in Alzheimer's with aging and start to address those. And the way we're going to figure out each individual's risk factors and the treatments will be through biomarkers, just like in cancer. And so that's really exciting. That's why we're so focused on biomarkers today, because we're going to have to type each patient's biology and then tailor drugs to their individual biology, which points out the indispensable role of the biomarkers. Mark mentioned our diagnostics accelerator. We actually are very proud to be able to say that we were very early investors in helping to bring the first 2 and only diagnostic tests to market. This PET amyloid scan, which is on the market with, you can go down to your radiology office today and get a PET amyloid scan, usually the one that's provided by Lilly. And you can find out, in my practice, I can tell the patient within 24 hours with about 90% certainty whether or not they have Alzheimer's disease. And I personally find it an extremely useful technology. It's expensive, it's a little invasive, an IV infusion, not a big deal. But what we have on the market today in 49 states, and we're waiting for New York to approve it, is a blood test. And the blood test is getting better and better. It's being commercialized by a company called C2N that we invested in very early, about 10, 15 years ago, spun out of Washington University. And literally today, a doctor can order a blood test and tell someone with, again, about 90, 95% certainty whether or not they have Alzheimer's. And that's really a revolution, not just in clinical care, but also in clinical trials, because it's really made clinical trials much more efficient and less effective from the point of view of screening and monitoring treatments. And the really exciting thing, I think, is that we have many more biomarkers in development. So the same company, C2N, that put out the first beta amyloid blood test is going to come out with a tau test that is reflective of the tangles in the disease. And that's probably going to be an even better test. And we're working on blood tests for inflammation and other areas. So let me just conclude by saying that our team at the ADDF comprised of 11 full-time neuroscientists that are constantly searching the field for the best opportunities and vetting them and working with companies to help them design their Alzheimer programs. So we play an active role. Our venture philanthropy has paid off in many ways to the foundation in terms of returns on investments. And you're also going to hear today about how the catalytic funding that we provide also helps investigators gain government funding, which both of the people here and Dr. Lexinger also has received many, many tens of millions of dollars, multiples of the kind of funding that we've given. So with that, I just want to briefly introduce Dr. Gary Gibson, who's a professor at the Brain Mind Research Institute at Weill Cornell. He's a world-recognized expert in metabolism of the cell, and we worked with Gary for quite a while, I think, to help him design and conduct his clinical trial of a supplement, a novel supplement for the treatment of Alzheimer's disease. I think it's pretty fair to say it kind of worked. And that was recognized recently by a very large investment from the government to help you advance your work. So with that, let me turn it over to Gary.
Thank you very much, Howard. I want to thank the organizing team for allowing me to present to this prestigious audience. All scientists love to talk about their work, and so this gives me a chance to talk about it. But I'm really just a spokesperson for a very large team that we endearingly call the Benfo team. And Jose Luxinger, who isn't here, is also a critical part of that team, as well as the metformin team. And I also want to thank Howard and ADDF, because without them, the story that I'm going to tell you would not have happened. Our brains use lots of glucose or sugar, and it uses it for many things. And in Alzheimer's disease, the ability of the brain to use glucose is abnormal. The dogma in the field has been for many years that the reason that the brain of an Alzheimer patient can't use glucose as well is the plaques and tangles are killing the brain. And so then the glucose utilization goes down. We have long taken a very different viewpoint of this. We think that the change in the way the brain uses glucose is causing those plaques and tangles. So one might wonder how you could see a connection between. So we think that the glucose is a therapeutic target. Glucose use in the brain is very complicated. So choosing a therapeutic target is complicated. 3 critical steps in the use of glucose in our brains require the vitamin B1, which is called thiamine. And we have measured thiamine-dependent processes in Alzheimer brains, and they're diminished. And we don't think of this as a general dietary deficiency at all. We think it is a brain-specific deficiency of thiamine. And you might ask, well, how could that happen? Well, thiamine is transported into the brain, and the ability to transport thiamine into the brain is diminished in Alzheimer's disease patients. And recently, people have surveyed 1,500 common drugs, and 20% of them interfere with the ability of the brain to take up thiamine. So that's where we think that thiamine deficiency comes from. In the lab for many years, we've studied the effects of thiamine deficiency on models and animals. If you make an animal model thiamine deficient, you create a lot of Alzheimer-like changes. You cause plaques, you cause tangles, you cause inflammation, you cause neuron loss, and you alter glucose metabolism. On the other hand, if you take a drug called benfotiamine, and it raises thiamine 100 times normal, If you give that to those experimental animal models, you diminish plaques, tangles, inflammation, neuron loss, and you improve memory. This suggests that this is an appropriate therapeutic target. Benfotiamine is a very safe compound. Like metformin, it was used in some diabetes trials. In Germany, they did a trial with benfotiamine. There were no side effects at all. The benfotiamine that we give to the patients is made according to FDA standards. So all of this preclinical data then suggested to us that we should test this in Alzheimer's patients. So we did a single-site study at Berkeley. We included 70 patients, all of whom were Alzheimer's patients, so half were a control or a placebo and half were benfotiamine. This confirmed that benfotiamine is extraordinarily safe. There were no adverse events. They were treated for 1 year, no adverse events from this compound. And there were very encouraging clinical results. There are some common measures of neurodegeneration that people use. One of these was diminished by 44%, one by 77%. So remember, this is a small trial, but these results were very encouraging. So taking the preclinical data, also the pilot trial data, we approached, and that included Jose, who isn't here, we approached the Alzheimer's Disease Consortium Center at UC San Diego to write a grant with us to do an expanded trial. That grant received a very good score, and the $45 million in funding started this past July. That will allow us to do a national trial with as many as 50 centers and well over 400 patients. And in this trial, we will double the dosage of benfotiamine because the preclinical studies suggest you can go much higher. And we will, of course, do the very best monitoring of the safety for the patients, even though we think it's very safe. We will, of course, use the very best ways to measure the status of the Alzheimer's disease for cognitive testing. What is most amazing to me and very exciting is what Howard referred to before. With the use of the biomarkers that Howard referred to was only a dream until very recently. What we will do in this trial, and it will be one of the first in the world to do that, because these methods are now so well documented that we can use them in the clinics. We will use blood biomarkers to measure plaques. They will be a reflection, I should say, of what's in your brain, of plaques, of tangles, of inflammation, of neuron loss, and abnormal use of glucose. All those things that we discovered in the animal models. We will use these both for diagnostic purposes, but also to follow progression along with the measures of cognition. So in my view, this is like science fiction. We've dreamt about it so long. I would also like to go back to what I mentioned at the beginning about the role of ADDF in funding the pilot trial and helping us go forward. It wasn't that we sent a great grant to ADDF and they said, here's your millions, do your pilot study. Howard personally spent many hours with me and the Benfo team designing ways to get funding of a compound that has very little intellectual property. And obviously that helped. So, and I also want to reiterate that scientists always like to talk about their science, so please feel free during this conversation or afterwards to ask me questions or even email me. Thank you very much.
Thanks. So benfotiamine, people can buy it on Amazon, right? And actually, I've used it. I've recommended it in my practice because, as you say, it's safe and makes a lot of sense. So if you go on Amazon, you see a lot of different preparations of benfotiamine and a lot of different doses. And it's sold as a supplement, I guess, which I suppose one could categorize it as a supplement in some ways. So in a way, it's kind of an example of the use of a supplement. And people, I think what's really important, as you said, is that, we all know vitamin B1 is in every multivitamin that everybody takes every day, but this is like a much better vitamin B1 or thiamine type of drug. I wonder if you have any thoughts about which benfotiamine, if people want to take, and also, should people in the audience be going to their doctor based on the data that you have and saying, I heard Dr. Gibson, an expert in mitochondrial diseases worldwide known, has recommended or has done a study to show that benfotiamine is valuable. Should people be taking it at this point?
Yeah, so this is a very slippery slope, and it takes a very wise person to deal with this slippery slope. First of all, with any food supplement in the US, you have to realize it's not regulated. The very famous case within the last 2 months was a congressman from California whose wife was taking mulberry leaves for weight control, and it killed her. They don't know what's in mulberry leaves. The benfotiamine that we used in the trial is not available commercially. We know everything about it. And with ADDF's help, then we can move this, and in terms of approaching NIH or companies for funding, the nutraceuticals treatment people are very skeptical of it. So that's the one side of the slippery slope. The other is, this is available. I certainly believe our data. And so then how do you decide? And this is, I think this is what's been called a shared decision between you and your physician. The data we have, I believe in. We've spent a lot of time being as careful as we can with it. And you can buy this compound. When we initiated the pilot trial, I got two emails from different places, from people who ran websites related to benfotiamine. One was very upset with me. She said, if your product works, I'm out of business, because she didn't know the quality of hers. The other one was very happy, because then if you have a trial that works, then it justifies using their product. So on a group basis like this, there isn't one of these products that I can recommend. I stand by the results. If you give pure benfotiamine, there are no side effects. And so this is a slippery slope that the physician and the patient have to deal with themselves. And since there are no data on these, I think somebody should at least do due diligence and see if any one of them have published safety data. So this is one of the reasons that I have such great respect for Howard. He knows everything I told you, but if you are afraid of moving forward, you don't go any place. And so this is a very important shared decision.
Mm-hmm. So I wonder if the examples of the role of thiamine supplementation or replacement in alcoholics who are in a very severe state and they're thiamine deficient, and then you give them the thiamine in the clinic. I mean, I've seen that.
Yeah, so this is the thiamine. I left that sentence out, sorry. Thiamine has been known since the 1930s, thiamine deficiency, to cause severe memory deficits in humans. And if you catch this early enough, you can reverse it with thiamine. If you wait until the the neurons have died, it doesn't work anymore. And there are many similarities between thiamine deficient humans and Alzheimer's patients. People have actually compared the memory deficits in both of them, and they're similar. And if you give thiamine, no one, to my knowledge, has given benfotiamine to these patients. They're called Wernicke-Korsakoff patients. But if you give thiamine to them, you can see an increase in blood flow and increase in cognition, as long as you do it early enough. And I think that's a good model for Alzheimer's disease as well. In general, and we looked at the data for general supplements with vitamin B1, and what we're giving to the patients is much, much greater than that would be. We were concerned in our inclusion-exclusion criteria if we should say, well, they can't be on any vitamin B1 supplements. But if you look at the data on the bottle, the amount of thiamine that you get from those supplements is trivial compared to the amount of thiamine that you'll get with benfotiamine. So the benfotiamine supplements, I think, are worth considering, but very wisely.
And what kind of dose do you know? I think people probably in the audience are seriously considering whether they should, people are desperate and they want to do whatever they can, especially
I agree. So
since it's safe.
the dosage that we used in the published trial, so our pilot trial is published and it's public information. We used 2 times 300 milligrams. So you take 300 milligrams in the morning and the night. In the animal studies, it suggests you can go twice as high and get twice as good a benefit. We chose that dosage initially because the safety data from the benfotiamine and diabetes studies, that was the optimal dosage. So we're going to try a higher dosage, and the group at UC San Diego has a very clever design. So initially, all patients will be started at that higher dosage, and if there are any safety problems, and of course we'll monitor these really carefully, if there are any safety problems, we can go down to the dosage that we used in the pilot trial. Our guess is we'll be able to test this higher dosage, and that will also allow us for it to be approved by the FDA much faster.
So in terms of approval by the FDA, so right now there's a paper in the medical literature, which obviously most of the people in the audience are not going to be aware of or read or understand perhaps. And you mentioned that there's no intellectual property, I guess, around this at this point. So, intellectual property is not just a commercial type of thing where somebody's going to profit, but it's kind of the only way that a company could market this and sell it with FDA approval so that it can be disseminated to the wide audience of people in the world who want to treat or prevent Alzheimer's disease. So how do we get to intellectual property with what we're doing?
So we do have, that was also, I didn't use any cards, maybe I should have. We also have a patent pending with Cornell and Berk there are delivery methods that we can change. We also have new ways that we think we can do this even better, and I think that's which we will approach ADDF about. I think that's where the real intellectual property, the patent we have pending, deals mostly with methods of delivery and also the timing and the dosage. And it's fairly far along, but whether it will be approved, there is also a Chinese group that's been encouraging the same kind of thing, and they have a very extensive patent on a lot of derivatives and things. We include some novel ones that a medicinal chemist at Cornell came up with as well.
So actually, this is an example of something that ADDF is very interested in, and it's called, I can't see that. So it's the notification that our time is up. So I can't see, the staff is trying to, I got 6 minutes. Okay.
Yeah, the lights are too bright.
So this is an example of what we call repurposing or trying to bring supplements. You can see with a relatively small amount of money, ADDF supported the clinical trial. We helped you to design it, I think it's fair to say, and to execute it. But then, how do we take that from a paper in the medical literature to something that you all could take and hear about and all that through intellectual property and commercial development? That's something that we're really interested in because we've funded about 20 of these kinds of repurposing and supplement type of projects. And I think it's a very important role for the philanthropy to do it. Now, see, it's 1 minute, right? Okay. All right. But I just want to, sort of from the big picture, say that this is something that's a really important thing for philanthropy to do because big pharma is not going to do this kind of thing in all likelihood. And investors aren't going to kind of start off and say, yeah, let's test a vitamin and see what happens, But there was very good rationale for this for this project. And we're very pleased that, a couple $1 million, I think it was, that we used as seed capital to get this program going has resulted in Gary getting a $40 million grant from the government and start him down a commercial road so that this very exciting supplement can be turned into some sort of drug that can be used and disseminated for use among the, not only for treatment of people with memory problems, but probably for prevention as well. Okay, I guess my time is, thank you, Gary. All right, great, thanks, Gary. So I'm tasked with replacing Jose Luxinger here, which is not so easy for me. But let me give you a background. It's really actually a spin-off of what Gary's just talked to you about. So the brain is about 3% of the body weight. But at any given moment, it uses about 20–25% of the energy that is available to the body, mostly through glucose, but also through oxygen. And if you think about how, a good example clinically of how totally dependent the brain is on glucose, a diabetic who takes too much insulin and drives their blood glucose down below about 60, what's the first thing that happens to them? The first thing that happens is they go unconscious, on the floor, unconscious, because the blood sugar is too low. And the way to save that person's life is actually to give them a lollipop to suck on or some form of glucose. And then they perk up like this. This happened to my father many times. And that's just one example. But imagine this happening on a chronic basis. So diabetics get what's called insulin resistance. And insulin is critical for glucose utilization. And that's why diabetes is such an important cause of chronic disease, retinopathy, kidney disease, vascular disease, and has been shown in the last 10, 15 years to be a risk factor for Alzheimer's disease. What's the link between type 2 diabetes, type 1 diabetes, insulin dependent, and Alzheimer's disease? Well, it's probably among many different mechanisms. In one part, the insulin resistance in the blood-brain barrier to the ability of the brain to use glucose efficiently. And as brain cells that are highly dependent on glucose for energy to work become deprived of glucose, they're going to start getting dysfunctional and ultimately die and form tangles and maybe make excess amyloid. So this has led to a number of therapeutic programs that ADDF has been interested in. And all of them are centered around the idea of using diabetes drugs, repurposing them for Alzheimer's on this basis of improving the insulin resistance that occurs with aging and in Alzheimer's disease. In fact, some people have even called Alzheimer's type 3 diabetes, which I think is probably overstated. So we've tested a few drugs, and I'll give you two examples. One is a drug called liraglutide. We seeded a large clinical trial at the Imperial College of London a number of years ago. Liraglutide is one of the leading drugs for for diabetes, type 2 diabetes, and it improves insulin resistance, so it improves the ability of the brain to use glucose. When we seeded that, the manufacturer of liraglutide came in and put several $1 million into the trial to make it bigger and more robust. I think we initially invested about $750,000, and then General Electric came in with the ability to measure beta amyloid in the brain and do some other things. And long and short story is that by seeding this trial and getting larger partners from industry involved, that we were able to, the investigator, Paul Edison at the Imperial College of London, was able to show that liraglutide actually helped in slowing down the rate of cognitive decline in patients with Alzheimer's and improving the biomarker profile of these patients. As a result of that, Novo Nordisk is now, if anybody's watched the nightly news, you're familiar with the tune, Ozempic, okay. So, so Ozempic is a trade name for a drug called semaglutide. Liraglutide is injected, Ozempic is an oral pill, is kind of a better way to give it. And Novo Nordisk looked at the data from liraglutide, and now they're doing a clinical trial of semaglutide for people with Alzheimer's disease. So it's a good example of how seed funding from the foundation resulted in partnerships to conduct a larger clinical trial, and the positive results led to a big pharmaceutical company to now going after that indication of repurposing a diabetes drug for the treatment of Alzheimer's disease, the semaglutide. And it's very exciting because there's a lot of other data around that. Now, metformin, which is the drug that Dr. Luxinger was going to talk about, is the most widely used drug for the treatment of diabetes. It's generic, and it's not expensive. It's very inexpensive. And it also improves insulin resistance throughout the body, including the brain. And there's a fair amount of animal model data that metformin can be beneficial in many ways, including, I think, this insight that Gary alluded to and Mark mentioned, which is, you know, the plaques just, except in the genetically driven disease, the plaques just don't occur. The plaques are probably the result of neuronal dysfunction, spitting out this beta amyloid into the brain parenchyma, the brain substance, and leading to the plaque formation, probably, I think, as a secondary phenomenon. And then, as a result of that, as the neurons get sick and die, they form the tangles. So the plaques and tangles are the pathology of the disease, but not necessarily the cause. And there are probably many causes, and one of them could be this insulin resistance leading to neuronal degeneration and then the spitting out. And that's not to say that getting rid of the pathology might not be beneficial. We're seeing that now with the monoclonal antibodies to beta amyloid from Lilly and Biogen and Eisai and Genentech. And it's very exciting because we'll probably get some approvals. But the thing is, we want to get back to more of those proximal causes, like insulin resistance, which occurs with aging. And so metformin, we funded Dr. Luxinger about 8 years ago to do a small pilot exploratory phase 2A clinical trial of metformin for people with mild cognitive impairment due to Alzheimer's disease. At the time, he designed the study so that these people would also be obese, because then we knew that people who were obese had insulin resistance. And he published that study. It showed some positive results and trends. It was a small study, only about 80 patients. But it showed that there was a good chance that metformin could be used to treat Alzheimer's disease. And the other thing about metformin that's really interesting is that in the anti-aging world, where we're looking for sort of what we call pleiotropic, like universal, holistic approaches to find one drug that could treat aging, and in treating aging could treat the multiple problems that occur, the multiple diseases that occur with Alzheimer's, including dementia, including Alzheimer's disease. And long story short, because I only have 2 minutes left, is that recently, again, that seed capital that we gave Dr. Luxinger has resulted in him, and I'm estimating, I don't remember exactly the number, but I think we gave him, I think, $750,000 about 8 years ago to do that small pilot trial. He just got a $40 million grant from the National Institute on Aging to do a large clinical trial to definitively ask the question, does metformin slow down the progress of Alzheimer's disease in people with mild cognitive impairment? So we're very excited about that. And again, I know some people are going to ask me, you know, do I prescribe it for my patients, and what are the contexts to that? And for the sake of time right now, I'll save it for the question and answer. But if you want to ask some questions about how we might use metformin in clinical practice today, because one of the advantages of repurposing is that we're testing drugs that are on the market. We know their safety profile. And as doctors in the United States, we have the legal right to prescribe off-label drugs, but we want to find a commercial way to get this to be having an indication through intellectual property so that these drugs can be widely used in the community of patients that's out there. And without that commercial intent, it's likely that studies like metformin will likely go into the medical literature but not be widely prescribed. So with that, we've got two programs that we've told you about that are non-amyloid programs, and yet they're really interesting because they address the metabolic changes and the insulin resistance that occurs with aging, and are good examples of our strategy that we've had from the very beginning, which is to translate the biology of aging into new therapeutics for Alzheimer's disease. No, Lisa Riccardi is the Chief Executive Officer and President of a company called Cognition Therapeutics. And I don't know how many years ago it was, but I think it was about 10 or 15 years ago, if I'm not mistaken, or something like that.
A little longer, yeah.
A little longer, sorry. Yeah, we do stay in the game for a long time sometimes. When a scientist named Susan Catalano had an idea for developing a small molecule approach, an oral drug approach to treating the amyloid that occurs in Alzheimer's disease. And we were one of the first investors in Cognition Therapeutics many years ago. And Lisa is here to tell us about how this has evolved over the years and the progress and where we are now. I think the really exciting thing, which Mark also mentioned, is that the monoclonal antibodies that are out there now that are probably, I believe, going to be approved in one way or another in the coming months, all in, the total costs are somewhere between $50,000 and $100,000 a year. They're infusion products. They have serious side effects. They do get rid of the amyloid out of the brain, preventing the effects of amyloid and being toxic to neurons and being part of the cause of the progression of the disease. I think what's really exciting about Cognition Therapeutics, and I don't want to steal your thunder, is that this is an oral tablet, hopefully, that will be much less expensive and hopefully more effective because of the multiple mechanisms of action. So we're really excited about it. So
thank you.
If you want to talk about
Thank you for inviting me. It's a privilege to be here. Thank you to your staff, and hello, everybody. We will talk about the science, but first I want to tell you a story about why what you do with the foundation is critically important. As Howard pointed out, we had a brilliant scientist who started this company, a woman named Susan Catalano, perhaps well known to many of you here. She left the Bay Area to go to Pittsburgh because the cost of doing research in Pittsburgh was a lot less. 3 years into her study, here we have a small company, lab animals. We still have the lab animals. And she had a group of scientists, 3. These were people who lived at home. Think of your kids 2 years after college. You don't have to pay them a lot of money. That's the background of the early days of this company. When the company was 3 years old, they were given the first grant by the foundation, and it was a $228,000.
It was an investment, not a grant.
An investment. Let me correct my language. At that time, Susan and her co-founder did not have any publications. You try to get money in a scientific or even a technical field, and you have no publications, it's really tough. So this money was critically important. At that same time, what we received from the government was $13,213 from the government. Grateful for all of it. That grant was to study the prevention of oligomer binding and how it can mitigate downstream toxicity. Now, why do you care about that? Oligomers are the most toxic portion of the plaques everyone is trying to clear out of the brain. Let me tell you something else. Do you know what I talked to investors about last year, last week, and next week? The prevention of oligomer binding to mitigate downstream toxicity. I think this is so important. Here's an idea that has come through the IPO process, a great deal of funding. We're in multiple clinical trials. Where did it start? I would like to believe that without the support of the foundation, the commitment of Howard to what Susan was trying to do, we wouldn't be sitting here talking about multiple clinical trials. It's actually really exciting. And it all started from the most humble origins. We went on to get another 100,000 from the NIA in 2014, more grants, 2016 biomarkers. Boy, big credit to the foundation for the pursuit of biomarkers. Howard described things we're using in our clinical trials now. They didn't exist 10 years ago, 7 years ago. It was probably a good idea 5 years ago. That's how things evolve. It's really true. 2016, another $2 million grant. This was from the government. And then from the foundation, another investment to support a phase 2 clinical trial. And my observation, once you get out of the small dollars, early studies, preclinical models, rat models, it's a whole lot harder to get your hands on big money. So getting a grant like that, or excuse me, an investment in 2019 was tremendous. Now, one thing Howard and I talked about is the ecosystem for funding early companies, and it is true. Susan and her chemist co-founder would drive around to family offices. They were in Pittsburgh. They would go to high net worth individuals. They couldn't fly around to a lot of places. Now today, years and years later, people who are still in the stock, Maine Angels, Cowtown Angels, Tech Coast Angels, early stage seed investors. And part of what allowed them to get there was to say, well, we have these awards, we're making progress, we have investments. It's a cumulative process. I have a great deal of friends who are Irish. I served on a board of a company, and they always use this expression about hard yards. These are hard yards. You have scientific inquiry, you have some ability to pursue the science, and it is all about the money. It is all about getting money to pursue these ideas. And so creating that ecosystem was a result of the things that came before it, which included the funding that we're talking about, or the investment that we were talking about here. When I looked at the annual report last year, I think for the foundation, there were 125 companies who were doing work with clinical companies, diagnostic companies, biomarker, maybe even imaging companies. And the reason I bring that up is because I really want you to know it is true. That is where the inception of science begins. People think, oh, you're in Boston and Cambridge and the money falls off the trees because all the venture people are there. Oh my gosh, this is really hard to scale up. And yet, this gives rise to really successful organizations. You mentioned 20 of the things you funded or invested in have now progressed to clinical trials. We're among them and grateful to be there. Let me talk about one other aspect, and that's geography. So I have at 2 occasions in my professional life lived in Boston and Cambridge and commuted from this area here. And you can't beat the density in Boston and Cambridge or the Bay Area. And people in Boston are running around going, we are the Silicon Valley of healthcare. And I don't know what people in San Francisco, they say, we are Mecca. This is where everything happens, right? There are constructs, really important ones, about where science begins, particularly for life science. Now, why is that? Because that's where the venture shops are principally. And the venture capitalists want the companies right there. And it's all a model that works. I'm not knocking it. But if you're a scientist in Pittsburgh, you have no shot. You can drive your car there. You're not part of a community of scientists. You don't meet informally. You don't get to go listen to interesting talks and find other people who introduce you to other people. So I think an important part for our company in our relationship with Howard and the ADDF was getting put on the map, if you will. And when you're a scientist, you don't even know you need to do that. You're out of the fairway of the economics for this early stage funding. So I think that is critically important. It becomes a virtuous cycle. One thing I do want to talk about is cognition and what we're doing. Our lab is in Pittsburgh, and we really have scientific research going on there. And we do early assays. There are rats and there are scalpels and all kinds of stuff you don't want to think about. But the work came from this woman, Susan Catalano, and her co-founder, and it continues. So we're there, and we have people who are here in the New York area. The office I run is in Westchester. We took the company public last fall. We have 22 employees and another 20 who are an intimate part of the organization. And right now we have 3 clinical trials, phase 2 clinical trials going on in Alzheimer's disease, 2 of them, and 1 in a condition called dementia with Lewy bodies. Now, what's helped us very significantly along the way is that we have received from the NIA, which is a division of the NIH, over $170 million in funding. I will tell you, I talk to investors morning, noon, and night, and they're never anything but appreciative. That's a big number. It really is. And it's what has allowed us to demonstrate our credibility and to continue to do more work and to help private investors as well as institutional investors look closely at the company. You have to break through the noise. I think Howard referred to 120 clinical trials. It may even be higher. And it's hard. It's really hard work to get and maintain the interest of those audience. So the validation that comes with that kind of grant funding, which came from early philanthropy funding, that's all critically important. So we're running trials now. We'll start another trial later in the year, and this trial is for patients with very early form of Alzheimer's disease, 500 patient trial. This study alone was funded for $80 million. I came from Pfizer, let me tell you, nothing, it's like being in Texas. There's nothing small. When you're in an incredibly well-run, stellar organization like Pfizer, to me, $80 million is a lot of money for this trial. So that's the next trial that we'll run. And eventually, we hope to study a disease for the back of the eye that leads to blindness, dry, age-related macular degeneration. As Howard said, we're talking about an oral drug. Can you imagine that, an oral drug to treat blindness, not an intravitreal injection? That's down the road. Our focus today is mild to moderate Alzheimer's disease patients, then early patients, as well this population, patients that have dementia with Lewy bodies. These patients not only have the amyloid oligomers that Howard was talking about, they have alpha-synuclein oligomers, which often leads to things like Parkinson's disease. So we are in just a state of intellectual wealth in the work that we do, and privileged to have been supported to get here. And now the capital markets have to behave and allow us to keep raising more money. It never ends, but it's a privilege to do this work. So again, my thanks to you and to your organization, Howard, and thank you for letting me be here today.
Well, thank you for joining us, and I think for the audience, you're a great example of a bio-entrepreneur. And I'm serious that you're telling us the challenges that you face as a small company. I do remember when Susan Catalano first came to us, whenever it was, 20 years ago, and she had an idea. Mm-hmm. That was about it, and it was really starting off with the very basic, earliest stages of trying to figure out if she could find small molecules for her idea, and it's come a long way, and I know she's discovered new pathways for how the drug works, which was really exciting. So I guess Mark's getting up to take the next one.
That's so long.
And Howard, not to worry, because you're getting first question, and I know where you're going to lead that question to. So for folks in the audience, if you have questions, you want to just indicate it. We have folks with mics who are going to come around, and we'll have lots of questions, I know. We're going to let Howard start with the first question, because I suspect you have a question off of our last presenter, and then we'll bring it to the audience, and Howard will run all that. So go ahead.
Well, I just wanted to make two comments, actually. One is that recently, after 9/11, I worked with the Partnership Fund for New York City to redo the New York City economy and kind of push it away to evolve the New York City economy away from finance, and it was recognized by a group of city leaders that New York City has more academic institutions than just about any academic medical institutions, more than just about any city in the world, and we had more grant funding from the National Institute on Health than just about any city in the world, but we had no commercial biotech industry. If there was any nearby, it was over in New Jersey where pharma was. So there was an effort by a lady friend, Maria Gotch, who led this really amazing change. And so today, New York City has a very robust life sciences industry. So when you talk about Boston and San Francisco, I read something recently that New York City is actually potentially becoming the major hub of new company formation in the life sciences, which is really exciting. And you also mentioned something that we hear a lot of that's part of our model. That's, I think, it's kind of hard to imagine, but it seems to be true, that companies really value an ADTF investment, not just for the money, because it's not huge money, but it's for the due diligence that our 11 neuroscientists do, the value and the quality of the due diligence that we do that gives investors the confidence to be able to invest in small companies that are early stage and risky. And everyone knows Alzheimer's is a risky road to travel. But there is some benefit in not just the financial, not just the strategic, not just the planning and assistance with the design of the work, but also the kind of...
It's real validation. You're exactly right. It's exactly right.
Thank you. So any questions that we have?
I actually have two. I have one for Dr. Gibson, which is that you mentioned that there are drugs that we are taking that interfere with thiamine in the brain or that deplete it from the brain. And I just wondered if you would tell us what drugs those are or what classes of drugs. And then for Dr. Frillett, you mentioned three diabetes drugs. Which one do you think will wind up being more effective for Alzheimer's?
Yeah, so this is a wide spectrum of diseases. I don't remember specific ones, but like a lot of antibiotics interfere with thiamine, chemotherapies interfere with thiamine, almost every range that you can see, and then you can do specific drugs. And nobody did a screen until recently, and so now they can start figuring out which ones of these and which ones you can avoid. So there's a chemo brain that's really well documented. You can see flyers on this in the hospitals when you go. A lot of these are related to thiamine transport.
Yeah, in terms of the one to take, metformin is quite inexpensive. It's generally without side effects. I mean, some of the side effects are gastrointestinal, and people have to be careful about their hydration. And in my little practice here in the city, a lot of people ask me, it seems like metformin is the drug du jour these days. And quite frankly, I'm a little cautious among the drugs that I've been repurposing, so to speak. I've become a little cautious about metformin, and the way I've come about to be prescribing it for people with memory problems or a diagnosis of Alzheimer's disease is to do their hemoglobin A1C, which is the diabetes test. And if their result is obviously in the diabetic range, then it's very clear to be prescribing metformin for diabetes with the added potential benefit of preventing the progression of any memory problems that people have. When it's in the range where it's like what we now call pre-diabetes, I think there's also a good rationale to take the metformin, not only, again, to treat the pre-diabetes, but also for the potential benefit of metformin on the brain. When it gets to people, and I've seen some patients recently that have normal hemoglobin A1Cs, and they want to try something, then it becomes a more difficult decision because of the potential risks, I suppose, of metformin. So I've become a little more cautious, but I think it's potentially good. And I think one thing about metformin, and one of the issues is that the idea is that it's slowing down the rate of progression, so we can't really tell if it's working on memory because it's not really sort of a cognitive enhancer and it's not making people better. I think in terms of which diabetes drug I would prescribe, probably the metformin in the extended release formulation, but it's going to be really exciting to see what happens with Ozempic, because if that works and we have the data from a big pharma company going after an indication, then that's going to be the answer, because it'll be approved by FDA.
I don't know who should answer. I guess. What? Oh. Maybe it's too early to answer this question, but have you seen any research to show that COVID precipitates cognitive decline, particularly in senior citizens? And if so, how is it related to Alzheimer's?
It's funny, the second time I've been asked that in the last 2 days, but it just so happens that last week there was an article in one of the leading medical journals about this issue, about whether COVID could actually precipitate Alzheimer's. And more broadly, it asks the question, what's the role of viruses in the cause of Alzheimer's disease? In other words, there's many ways to get to the plaques and tangles. If you have a virus in your brain that's causing neuronal injury, then the expression of that at pathology is going to be plaques and tangles. So there is evidence that long COVID can be associated with long-term cognitive the fog that's described with COVID, and that can be long-term. And the theory that viruses that live in your brain cells for decades could be a contributing factor to Alzheimer's disease actually makes sense, but it's very hard to prove. Herpes simplex, herpes zoster, RSV, all these viruses that we live with every day that can live in our brain could be contributing factors, but we haven't been able to prove it. And then the other thing is, even if you believed it, and we could have biomarkers that an individual has, let's say herpes zoster in their brain, or herpes simplex, or COVID, coronavirus in their brain, how would we treat it? I think that's the other question. What kind of antivirals would we give to treat that. And there are a couple of companies out there that are pursuing this kind of approach, but I think it's hard to prove. I think the viruses could be a contributing factor, but probably not the sole cause of Alzheimer's disease over the course of a lifetime.
There's a social dimension as well. In one of our trials, we ended up determining that we needed to enlarge the trial. Why? Because you have elderly folks, you have isolation, all of these things that went along with us being in quarantine for months and months. So it's not the same thing as a bacteria or virus, but from where I sit, I would say it has made an impact because it made it harder for these folks. People didn't get to treatment, and so we ended up over-enrolling in a study to combat some of the social consequences of COVID, which is different than what Howard is talking about, but still has a cost to those patients and families.
And I think that's really important because probably a lot of these people, and we learned this from post-operative cognitive decline, a lot of people are undiagnosed. They have Alzheimer's, but they're undiagnosed. And then they just kind of go along, and then they get COVID, and suddenly they're worse, and it's obvious. And then they get diagnosed, and then the conclusion is the COVID caused the Alzheimer's. But probably before COVID, they had cognitive impairment that was unrecognized.
Also, I'd like to mention there is a whole ward at Weill Cornell where a physician, that's her main practice, is treating long COVID. Some of those patients, COVID has a wide range of symptoms, but some of those have the same symptoms as a thiamine deficient human would have. And there are other viruses that actually do interfere with thiamine transport between different parts of the cell. So I think long COVID, a lot of elderly people, in Germany the numbers are as high as 40% of elderly people that are sick, that are in a hospital, are actually thiamine deficient. So I think that there is certainly a long COVID, and there's a large number of physicians treating them, and they have cognitive deficits. Some of those could be related to their metabolic changes as well.
My question is about the blood test. And right now it's for Alzheimer's, and I assume that what it tells you is the amyloid in the brain, correct? So is it possible that that could extend for Lewy body dementia to the alpha-synucleins?
Yeah, so the alpha-synuclein is the equivalent of beta amyloid in people with Parkinson's disease and in people with what's called Lewy body dementia. And through the diagnostics accelerator that Mark mentioned earlier, which is our $100 million program to develop new blood tests and eye tests and speech and language as early diagnosis, we do have a program for alpha-synuclein blood test. And I think you're going to see that in the next year or two. It turns out that measuring alpha-synuclein in the blood is harder than measuring beta amyloid or tau in the blood. But I think it's going to happen. And what's enabling that is new technology that is so highly sensitive. You know, you can kind of see 1 part per billion now with the new technology, and that's really it's the new technology that's revolutionizing the kind of blood tests that we can develop. And so the alpha-synuclein in the blood is a very, very small amount, but it's measurable, and we're starting to see companies, biotech companies, looking at alpha-synuclein, so it's a great point. And it also points out that we're going to see many different blood tests for Alzheimer's, the same way that, again, in cancer, we type patients' tumors. I think we'll end up typing people for many different blood tests that characterize each individual's disease.
Do you have any time frame on this?
I'm sorry?
Do you have any time frame on this, especially for the Lewy body portion
the
of it?
alpha-synuclein, I think we might have proof of concept in the next couple of years, and then commercial development might take a couple more years, so maybe 3 to 5 years.
There is a skin biopsy. We're actually using it in one of our clinical trials, a punch biopsy. You go to multiple sites in the body, and it's a bit of a lottery. If you have 1 site, it's Parkinson's, 2 sites, it's multisystem atrophy, 3 sites. You unfortunately punch the ticket that says DLB. I'm not sure if I've gotten those stated in the correct order, but as Howard said, they're not nearly as sophisticated as what he is describing that is in the future, but it's an evolutionary process. A lot will be learned in our trial, just gathering patients and using this form of testing. And then that knowledge will help feed into other tests going forward.
Yeah, and what is being brought out here is Lewy body dementia. So it turns out that people don't just have pure diseases, generally speaking, pure forms of dementia. Elderly people have what we call multimorbid pathology. So we typically even today, we've only talked about plaques and tangles, but when you go to autopsy in people with Alzheimer's disease, it turns out that 35–40% of them have Lewy bodies as well, and then they have other problems. So, you know, it's really what you were alluding to, it's these misfolded proteins that are occurring in the aging brain, and that leads to other novel strategies for trying to treat by enhancing the cell's ability to get rid of the misfolded proteins in general. But we need the biomarkers to characterize people because that also affects treatment. If we're giving someone an anti-amyloid antibody where the person also has Lewy bodies, then we're not treating the Lewy bodies. Any other questions for us? This one here.
This is just a matter of curiosity because you've spoken a lot about diabetes and Alzheimer's. Like, is there data in terms of the overlap between patients who have Alzheimer's and were also type 2 diabetic?
There's a lot of epidemiologic data showing that diabetes is a risk factor for Alzheimer's disease, absolutely. And there's data looking at people with diabetes and in terms of their cognitive decline, rate of cognitive decline and dementia. So I think it's pretty well accepted now that diabetes is a risk factor for dementia and cognitive decline and Alzheimer's disease. Does that answer your question?
Well, I was just more curious, do you know the percentage of people who have Alzheimer's who have type 2 diabetes?
Well, about 30% of people over 65 have type 2 diabetes. And let's say about 25% of people over 75 have dementia. So we're talking about two very common illnesses that co-occur.
This is for Lisa. Can you give us a little more detail or description of the trials that Cognition is doing?
Be delighted to.
Some sort of time frame?
Yeah, absolutely. Thanks you for that question. The first trial we call SHINE. This is a trial with 140 patients. The patients are mild to moderate Alzheimer's disease patients. We began the trial. 2020 comes, can't get patients back to be tested, no follow-up, shut down sites in Australia where we were doing work, and then we received, a year later, another $16 million to finish the study. So it's ongoing right now. So as I said, the criteria are mild to moderate patients, and we're recruiting outside the US for patients in these trials. Surprisingly, it's challenging. The patients are more reluctant to go back into an institutional environment to participate. There aren't people working at the study centers. I joke with investors, how long do you wait for pizza at your local pizza shop? You can't find the 17-year-old kid working there, right? It's not different in other parts of our economy. So the trials are somewhat slower for this reason too. So that's the first trial, and those are mild to moderate patients. The second trial that's ongoing right now is a trial, we refer to it as SEQUEL, and what we're looking at there are brain wave patterns. So this is a trial that is 4 weeks of dosing, a washout, 4 weeks of placebo, two different arms. They're each doing something different at the same time. And what we're looking to see is if treatment with CT1812 targeting the oligomers, as we do, allows brain wave patterns to return to normal. These are disrupted in patients with Alzheimer's disease. I look at this study, and I suspect Howard might agree, and Gary as well, as somewhat of a biomarker study. It is unlikely you're going to see a change in efficacy in patients with mild and moderate disease in 4 weeks. I mean, that would be very surprising and a wonderful thing to come out of it. But what you want to look at is something indicative of whether you're having an impact on disease. So that's the second trial. It's nearly done. The other trial that we're recruiting for actively right now, we have over, I think we probably have 20 of our 30 sites in the US. And this is for patients, as you were alluding to, who have dementia with Lewy bodies. Now, the interesting thing here is this is also a patient, we have a study with 120 patients. These are both 6-month studies. It's challenging for the institutions. I was in Texas last week visiting a premier center, and they said, oh yeah, we see 5 patients a month, and I'm thinking, thank goodness we'll get this trial recruited quickly. These patients come in, and they said to me, Lisa, 90% of the time, patients come in, and it happens to be a bad day. What does that mean with this population? Terrible interruptions of sleep, which affect your cognitive ability, hallucinations, small children, furry animals, GI disturbances. Dementia with Lewy bodies shares the cognitive impairment, but has a whole series of other conditions that the patients suffer from. It's very diffuse. It goes on for for a long time, typically 18 months, 3 doctors, and 7 visits before somebody goes, holy cow, look at all the alpha-synuclein here. You are a patient that appears to present with dementia of Lewy bodies, dementia with Lewy bodies. So that's the 3rd trial that's running right now, 6-month study, also 140 patients. The next trial is 540 patients at 50 to 60 academic sites around the country. And we hope this fall, December, to be opening those sites. So this is an earlier patient population and a much longer trial. Why? Because when you recruit early patients, the frank matter is you have placebo patients and drug patients, and you have to wait for the placebo patients to frankly show more of the disease. That shows the beneficial effect of taking drugs. So earlier trials are typically longer trials, and it's powered for a much larger group because you're going to see possibly less great differences in the patients. So all of that implies large numbers and a longer duration. So those are the programs going on now. Did that answer your question? Terrific. It's all on our website.
Can you quickly explain Lewy bodies?
So if you think of plaques, Alzheimer's described plaques in 1906 using a dye, but it wasn't until 1984 that George Glenna literally took the brains of people with Alzheimer's disease at autopsy, put them over a nylon sieve, which was tiny, tiny, tiny little holes in it. And the plaques, these round balls, the microscopic round balls, sat up on the nylon sieve. And then he took those and he isolated the main protein in the plaques, and it was beta amyloid. So now they're beta amyloid plaques. There are many other proteins in the plaques, but they're predominantly beta amyloid. Lewy bodies, you see them under the microscope in patients who are clinically diagnosed with this unique form of dementia characterized by sleep disorders and other things. So you can clinically make a diagnosis, but to get to the pathology, obviously, have to go to autopsy. When you see the Lewy body, a Lewy body is a pathological feature of the disease. You see it under the microscope. And the same thing, Lewy bodies were isolated, and alpha-synuclein was determined to be the main component of the Lewy bodies, and that led to therapeutic strategies to try to remove the alpha-synuclein or somehow treat it. The Lewy body treatment strategies are much more delayed than the Alzheimer's strategies, and cognition therapeutics is probably having one of the leading programs today in the treatment of Lewy body dementia, which is a significant part of the whole group of people with dementia, under-recognized for many years.
Absolutely.
So
by the way, the Lewy bodies are throughout the body. It's really shocking when you study it. Part of the reason these patients have such tremendous GI problems, it's diffuse and pretty significant. We were given a $30 million grant to study this condition, and as our PI in Florida says, Jim Galvin, this is the biggest dementia you've never heard of. To your point, Howard, it really hasn't been on the radar screen. I have met Robin Williams' widow, and she is a lovely woman. If you remember, he died in 2012. He took his life. And I said to her, how on earth, you're in the Bay Area, you know how far Stanford is from their house? Less than it took you all to get here today, right? It took all that time, and he was never diagnosed. To Howard's point, it's not well understood, it's not well clarified. You go to a bunch of doctors, each of whom wants you to go to another doctor. So anyway, bringing awareness to that condition is particularly important. I think Alzheimer's is on the map. Who can raise their hand in here that says, I have no loved one that's ever encountered Alzheimer's disease, right? Okay, 2 hands go up. A lot of awareness of Alzheimer's disease. Dementia with Lewy bodies remains, as Howard said, not so well understood or diagnosed.
Well, I can say from clinical practice point of view, it's hard to diagnose it.
Yeah, no, that's right.
It's actually quite hard. So I think we need biomarkers. We need these diagnostics.
Absolutely.
Howard, any last word you want to give just overall?
Yeah, well, I want to thank Gary and Lisa for joining us today, and all of you for joining us and working with us. And I think the panel today is representative of the progress of the field. These are 3 clinical trials in human beings of novel drugs. That's really exciting.
Yeah, and I just want to kind of glom on to what Howard just said. In Alzheimer's right now, 130 plus clinical trials that are going on. About a quarter of those are in the amyloid, remove the plaques and tangles, 3/4 not. I bring this up because it is extraordinary that we get to sit here today, lucky us, to get the insight on all of those trials and the specifics on 3 of those trials. And this is a way of saying, this is the cutting edge of the science that's going to come and take care of cognition and dementia and Alzheimer's. So down the road, and I'm going to come back to down the road, our loved ones are not robbed of their humanity. And I hope you all leave thinking about that today, because I can assure you of a couple of things. You're going to hear in the next year, it's going to be in the next 2 months, more and better on the amyloid drugs. You're going to hear it. It's coming. It won't be the be-all, but it'll affect a certain part of the population. And these other trials that you're hearing about, the complementary around metabolism and around insulin and diabetes and vascular, genetics, we're going to hear in a 3 to 5 year how those are starting to be combined, right? So that's the insight we've gotten today. How about a round of applause for fabulous? And this is where I excuse you all to exit because lunch is about to be served. Yay.